I thank the duo of speakers. The next talk is going to be by Professor Tzvi Bentwich, and it will be on the microbiome core facility. So, Tzvi, please.
Thank you very much. Because of the constraints of time, I may pass it fast, but I have to say, okay. Sorry. But I have to say that I am giving this lecture focusing, as you can see, on the microbiome core facility, which was established in this place actually not very long ago, between 1 and 2 years. And again, I am representing mostly 3 main investigators that I'll name in a minute. But before that, because most of you are not familiar with the situation here, I just want to give the credit to the Arava Dead Sea Research Science Center that I'm part of. I'm actually the scientific director of this branch, of this Masada. And needless to tell you that we are, if you visited us a year ago and you saw the cramped area and rooms that we were in, you can imagine that it is much more than a dream come true. Having said that, I just want to point out that this research center is actually a combination of 4 sites of R&D, historically science ministry supported. And just to give you an idea, we are talking about a group of about over 30 scientists with over 40 research assistants. And again, to give you an idea, when I came here 5 years ago, there were between 2 and 3 investigators, and now there are 10. There were no research assistants, now there are about 10. There were no graduate students, now there are about 3. So it has grown significantly, and what I am representing is just a small piece of the work that is quite impressive considering the number of people and the resources. Lastly, in terms of the research center, the vision is to put together healthcare R&D in the Dead Sea. You heard something, and I will mention that, but an emphasis on the application, on the applicable, on the applied science and research. That being said, one major group, which is now consisting of at least 3 investigators and an associate and research center, research assistants, is what we call the Skin Research Institute. And again, time does not permit, but I would want to emphasize that the platform that is developed here, and actually was initiated by the speaker after me, by Dr. Zevi Maor, is based on the skin organ culture. And this is a unique situation. There are not many laboratories at all in Israel and worldwide doing that, and therefore the results and the potential is very big. So, having said all that, I'm now focusing on the microbiome core facility. And as you can see, there are 2 avenues, so to speak. There is the environment and agriculture, which I'll talk in a minute, and then followed by microbiome, and more specifically by skin microbiome, which is dealing with humans. Microbiome, of course, is a buzzword, but I think most of you would know that microbiome now is the very, very hot field in biomedical sciences, because as we realize, microbes that I, as a physician, a trained physician, learned and grew up on the idea that only bad microbes that you have to kill and to eliminate, are the core and focus of medicine. And it becomes very clear that that is a wrong concept, and that microbes are all over us. They are what we would call the good microbes, or the balance between the bad and the good. And moreover, agriculture and everything around us is affected by microbes. So the first part of, and that would give you an idea. We have a very up-to-date, I would say, advanced methodology and equipment that enables us to do the very advanced type of the next generation sequencing and what is involved in microbiome studies. And the people and the leaders of this endeavor, to the left is Dr. Amir Schittenberg, in the middle Dr. Ashraf El-Ashhab, and in the right is still not a doctor, but almost a doctor, Michael Brandwine, who is a graduate student between us and the Hebrew University. And the topics that they are dealing with, is, as you can see, is the sinkholes, atopic dermatitis, which I will talk, nematodes down there, fishes above there, and acacia on the right. So I will start with agriculture and environment. And this would just give you a taste of what we are talking about. Dr. Ashraf El-Ashhab, in collaboration with I'm not saying exactly the institute, the Lakes and Sea Institute at Eilat, where they grow fishes, performed the following intriguing experiment. You can see on the left fishes that were subjected to exposure to pathogen bacteria, and the question was whether if you put them in tanks that the water has been exposed to UV radiation, and then you introduce the pathogenic bacteria, what will happen? And believe it or not, as you can see, in 24 hours and 3 weeks later, the tank that was irradiated, sort of purified by UV, all the fishes are dead. In the tank that was not exposed to UV, only 4 fishes died. So what that means is that practice, the usual practice of cleaning, so to speak, the water for the fishes is a bad practice. Because if you do that, you actually come out with a worse result. And obviously the question was what it is. And just to make a long story shorter, this is the dominant bad bacteria that dominates the fish that died. But the fish that didn't die, as you can see, there is the infection, but they are overcoming it. And the question that Dr. Ashraf asks rightly is it possible that these bacteria that you see here in light blue color, which then dominate the living fish, is it the cause of the protection? To make a simpler expression, a probiotic protecting fish from death. The time does not permit, so I will just conclude another study that he has done on Acacia. And the message to take home from that study is shown here in 2 illustrations. On the left are illustration of bacteria isolated from these acacia, which are not known, are not registered in the database, and they, as you can see here in the lower part of this slide, they kill fungus. And the translation of that is a field study in which those isolated bacteria are tested in their ability to protect peppers grown in a field from fungal infections. On the right, you can see another element that is intriguing, and this is the generation of growth hormone by Phyllosphere, by bacteria isolated from the Acacia. And there is a difference between two types of Acacia, but note the red bars. And the red bars stand for increased secretion of growth hormone by bacteria that are isolated from the Phyllosphere of Acacia. What is the message to take home, which I don't want you to read, but I would tell you the following. The extreme condition of the Acacia living in the desert select for very unique bacteria that have a potential of a promising application in the fields of agriculture. Now, I want to turn to the second investigator. This is Dr. Amir Schittenberg, and I will again only show you a taste. So he has been studying the sinkholes and the cultures or environmental bacteria that are living here. And again, the time does not permit to go too much into it, but again, the story is similar. Extreme condition, harbor and select for very interesting and intriguing bacteria. The major focus of Dr. Schittenberg's studies is nematodes. And just to make it clear, nematodes are one of the worst dangers, if you like, or problems that we face. It is estimated that it is $100 billion loss per year because of nematodes causing destruction of agricultural growth. So what Amir has been studying, this is a sort of illustration. When you look at the roots of these growth, you can see a normal looking roots. And here, the roots that are infested with the nematodes, which induce such a growth on the roots, which essentially kill the plants. And what comes out again of this study is that you have a novel bacteria. I would just point out one bacteria that has been isolated. Again, time does not permit, but they are singular in their characteristics, and it's a genus with a wide range of proven industrial applications. So, just to end up, the questions that are there are the bacteria in the nematode, in the what is called galls, that the growth on the roots, or in the soil to be concentrated on if you want to suppress or eradicate these nematodes. And this is an ongoing study. I would now turn to the last part, which is the skin microbiome. And as I mentioned, Michael Brandwan and the others are involved. And the study is, as you can see, there are several diseases. I should mention first and foremost, of course, Dr. Harari, who's in this audience. So we are looking at atopic dermatitis, psoriasis, anhidrosis, mycosis fungoides, folliculitis, diarrhea, inflammatory bowel disease, and chronic fatigue syndrome. And these studies are in collaboration with quite a number of institutions. And you will see also that there is an education part that is in collaboration with the Weizmann-Davidson Institute. So, to start with, atopic dermatitis, if you don't know, is a disease that is now affecting almost 30% of our children. It is a growing issue and a very serious issue worldwide. So what is the reason? The reason is that there is a dominance of staphylococci in the infected skin, while in comparison to the normal skin. And when you look at, again, I will just give you a taste, and because the time does not permit, you can see the following differences, which Michael and his team discovered. You can see that there is a very sharp difference but in ages. These are children, these are adults, and I'm sorry, these are teenagers and these are adults. So to start with, there is a very sharp difference, the meaning of which is not yet clear. The second element, which is again has not been noticed before, is that there is a dominance of Staphylococcus, the red, these are samples taken from the affected and non-affected skin of the patients compared to the healthy individuals over there. But again, coming to climatotherapy, there are the following points to be observed. This is the healthy, this is the moderate, and this is the severe. And I'm concentrating for that for a moment. And altogether, it is seeing the effect of climatotherapy, because we want to ask questions which are related to the area, which may bear on the use of climatotherapy here, and of course, ask a question which deals with the disease. And what you can see is very, very, very intriguing. There is a very clear, I would say, difference in the change post-climatotherapy in healthy. You cannot see it because of the relative scale. Yes, I'm sorry. But what stands out that in severe atopic dermatitis, to staph 1 and to staph 2, there is no change between before and after climatotherapy. However, the difference does appear in staph 3 and in micrococcus, and it does appear in all groups, that is healthy, moderate, and severe. Now, the message to take home, climatotherapy clearly affects the skin bacteria of both healthy and infected. Secondly, it affects people with atopic dermatitis, and there is a clinical correlate. And lastly, there is a difference between the different stages, and therefore the immediate question is, what bacteria can we replace the known bacteria at this time, again as an issue, if you like, of probiotic. The healthy microbiome is again a very intriguing story because there are apparently marked differences between individuals. And Mira shows me the time, so I will go fast. You can see only the difference in between 1, 2, and 3 volunteers in the prevalence or in the distribution of the bacteria. And this is a fungi, and you can see a guy here with a beard, and so you see the difference between him and him. So the time again does not permit. But I want to end with what is, I would call it maybe the most sexy and intriguing project that we are involved in. And that is what we call the Israel Skin Microbiome Initiative. And it is an initiative that tries to do the following, to create a data bank for skin microbiome which is non-existent at this time. And because we believe that we have a heterogeneous population in Israel, and if we can take about 50,000 samples that will be assembled it will be 100,000 biological samples. That is divided by 3 because it's from different samples, from different locations. So it's about 30,000 samples. We will cause the only skin microbiome biobank worldwide, but we will do it through the involvement of secondary or high school students and university students in an operation which would bring science to youth and at the same time create a very important and attractive and the reason is that there are differences between male and female. There is, of course, pregnancy. There is old age makes a difference compared to infants, exercise, heart, and of course, different ages and different ethnic background. So we believe that this is a citizen science project, and we are doing that with collaboration with Oded Kenan, Dr. Oded Kenan, who is leading the educational side, Michael doing from the core facility, and of course Davidson Education Center as a very important partner for making it hopefully beginning by location oriented and spreading out to the whole country. So I think I gave you a good idea. So that's the sort of, that's the idea that you have 20,000 citizens, 200 school students, 30 university students, 10 scientists, eventually, of course, and one database. So I thank you very much for your attention. Thank you.